is Dr. Stephen Varga. He's the Associate Dean for Academic Affairs and Grad Student Development and Professor of Microbiology and Immunology at the University of Iowa School of Medicine. And he's going to present for us on how to write a successful grant application.
Okay, great. All right, so I just want to thank you for inviting me to talk about writing successful grant proposals. I wish there was, you know, a secret I could share with you to be wildly successful at this, but there is no kind of one thing that you know, will really make you a successful grant writer. What I'm going to talk about, and I'm sure my colleague, Dr. Jaspers, who's going to present from the reviewer perspective, are things really to try to ensure that you avoid in your applications to make it easy for reviewers to pick at them. And so I just want to talk a little bit about kind of a lot of the important do's in grant writing, and then a few of the things you should avoid. So, and my apologies, this is mostly just kind of bullet pointed text as a means of discussion, and I certainly want to leave some time for discussion after this. So, you know, as you're developing your proposal, I think the first thing that you want to make sure is that your proposed work is going to be viewed as important to the field. So you want to develop an area that's going to move your particular field forward and is not going to be viewed as an incremental advance in the field. All right. So that's really important as you develop the novel idea you want to pursue. So I think, you know, you want to spend a fair amount of time working out your idea and how it fits into the field at large. And then you want to make sure you develop a strong central hypothesis and that you have the means to test that hypothesis. So it's great to come up with the perfect hypothesis, but if there's no technology available that allows you to test that hypothesis, it's going to be very difficult to write that grant. All right, so you need to make sure as you do this that your central hypothesis is one that you can test in your grant in the experimental approach. And then really an area that particularly for new investigators, I think you want to spend a lot of time on is focus on providing strong rationale in the grant for your central hypothesis and for each of your aims. That's really helpful to reviewers. You want to make sure you're citing the appropriate literature that serves as the foundation for the starting point of your idea and where you're starting from, as well as, you know, obviously, ultimately, you're going to provide them some preliminary data that serves as a jumping off point. All right. And then you also want to make sure that you're not singularly focused on your central hypothesis and develop this kind of tunnel vision. You want to make sure you consider the alternative hypotheses that could help explain any results that you're going to obtain in your experimental approach. And you really want to keep an open mind and not have a preconceived notion about what answers you'll get. Some of the best grants that I've read and that I think I've written are ones in which I don't already know the answer, or I don't strongly believe in one answer over the other. And that it really is independent of what answer I get. It's about asking important questions and then finding out the answers and going from there. So I think that's what makes, you know, for really interesting grant proposals. So as far as the specific aims, you want to make sure when you're writing them that they're really tightly focused and not too open-ended, and that they relate to the central hypothesis. So you don't want aims that go too far away from your central hypothesis. And in fact, I find it harder and harder to write grants that have more than 2 and at most 3 aims. Generally, grants that have 4 or more aims, you're sacrificing the rationale sections and the experimental sections, and in particular, the results sections of what you expect to get from those experiments, and that makes it easy for reviewers to deduct points, basically, because they don't know all the information they're looking for, because you can't simply provide it. You don't have enough words left in a 12-page grant. And so I tend to write, kind of two aim grants with lots of sub aims so that I can provide them with the necessary details. Sometimes I'll write 3 aim grants, but I like to keep it as simple as possible so that I can make sure I provide sufficient rationale and sufficient detail. Even, established investigators, when funding is tight and everything, and you don't necessarily always get credit for everything you've done, you still need to explain what you're doing in sufficient detail for the reviewers to understand, and you need to justify why you're looking at things. So because of that, and when a panel is reviewing your grant, only the 3 reviewers and maybe 1 or 2 other panel members, sometimes if it's in an area that they're interested in, have read your grant in any detail. When your grant comes up for discussion, most of the panel simply pulls up the specific aims page and actually just sits at that page during the entire discussion, unless somebody refers to a figure or something, on page X of your grant. Generally, most of the panel just kind of sits at that, or they'll scan through the grant on occasion, depending on how discussion is going. So the specific aims page is really the page that you can use to capture the remaining members of that panel that aren't the discussion leaders. And so you really need to capture their interest and imagination and really get them interested in what you're doing. So there's a bit of structure that I've found that a lot of successful grant applications utilize. I tend to use this structure, and I know many other applicants do as well. And that generally has about a 4-paragraph structure to it. The first paragraph is just outlining the problem and the current gaps in knowledge that exist in the field, right? It's really critical in 2 to 4 sentences to be able to synthesize the field and the background very succinctly, and then define the current gaps in knowledge that exist, right? And then the second paragraph is generally focused on what you're going to do to get at a solution to those knowledge gaps, right? And this is where you're proposing your hypotheses that you're going to test. And then the third paragraph generally outlines the specific aims that will test those hypotheses. And the fourth paragraph is basically the payoff paragraph of what your expected outcomes will be and the impact to the field. What performing this proposed research that you're outlining is going to do to move the field forward, right? And where will we be at the end of this 5 years? And that's a really important aspect of it. But it's those first couple paragraphs that really capture the essence of your grant, and you want to spend a lot of time crafting those and this entire aims page, because it's really important to really try to grab the attention of the reviewers. It's certainly, really good aims pages want me to read the rest of the grant. When you're a reviewer, you have, depending on the panel, between 8 and 12 grants to review. That's a lot of grants to read through. And so, the ones that capture your attention early make it easier to go through the rest of the 100 pages or so of the grant. So with all the budget information and everything. As far as the significance page, you really want to establish the importance of the work and where it relates to the field. So this again, if you're looking at various infection settings, obviously these have really importance to human health. So you want to make sure that you establish that and how your grant is going to help with those knowledge gaps that will move either treatments or vaccination or understanding pathogenesis forward for the field. And so that really helps contribute to the potential significance of the work. And again, just outlining how the research will advance the field. And then you always want to make sure that your work will have a long-term impact on the field and the research. Again, not being viewed as incremental, but moving it, a big step forward so that other people can build on it then in the future. Innovation is, I think, one of the hardest sections to write, actually, for many people, myself included. And it's also one of the most easily picked on areas of a grant. I think in general, an application can have one or more types of innovation. The two most common types of innovation that individuals spell out in their applications are the technical innovation that they employ and the conceptual innovation that they're providing the field, right? And I think it's the technical innovation one that really gets picked on the most by reviewers. And one of the most common comments you'll see on a grant application is, it'll always be in the weaknesses, that the applicant utilizes common approaches and is not technically innovative, right? I think actually, and I don't mean to offend anyone who writes that, but that's a pretty lazy reviewer comment, because every grant can't be technically innovative. We can't make up new techniques for every grant and have the grant solely be about exploring that new technique. You can use boring approaches and ask super interesting questions that haven't been asked previously. And so you want to make sure in your innovation section that you're explaining how you're using the techniques in really conceptually innovative ways. And sometimes you're using a technique that's been used a lot in one way, but you're asking a novel question with it. I would argue that's both conceptual and technical innovation, and you need to spell that out to reviewers. But again, this is an area that often people, even sometimes without putting a weakness down, they will give you a poor score in innovation without even defending it sometimes. You also want to highlight the translational or mechanistic insights that will be gained from your research. And it's important, again, that and NIH review panels are trying to highlight this, that your work does not have to be translational. It isn't required. It's not an NIH grant requirement. You can be looking at basic mechanisms of things. And so that should never be a weakness that is identified in your application. However, if it is translational, you should certainly highlight that. And obviously you want to make sure that you're proposing mechanistic studies in your research. And then also highlight any new theories, tools, or approaches that will be developed in your proposal. And those can be discussed in the innovation section. Providing strong preliminary data is critically important to any successful grant application. If you don't have sufficient preliminary data to support your application, it's likely to be unsuccessful. The preliminary data that you provide should support the hypotheses to be tested. They should lay the groundwork, for what you want to study, and you want to make sure that they relate to your aims and your central hypotheses. They can be a combination of both published and unpublished data. You can certainly put published data in as your preliminary data. That's not a problem. And you can use a combination of published and unpublished data in this section. And you want it to provide a proof of concept that establishes the technical feasibility of what you're proposing. If there's a new technique which you've never performed or that's very difficult because it's brand new, then ideally you'll have a key piece of preliminary data to support your ability to do that, or you'll have a collaborator that provides you a letter stating that they will help you with that technique. That's really important to establish feasibility. That's another common weakness that's identified in many grants, is that the PI is either unfamiliar with the techniques proposed, which can often come across in the writing of how they describe them and what they expect as far as the results. And so this is a common area that can be beefed up pretty easily. And you also want to make sure whenever you're showing preliminary data that they have the appropriate controls. Don't cut those out of your preliminary experiments just to kind of keep the graph size small or anything, because if it's missing the critical negative and positive controls, people will pick on that too. So just be careful about that. As far as the research strategy section and the approach, you want to make sure to articulate why you're taking the approach that you've outlined. That's, again, the key in the rationale section for the specific aims. You want to make sure that you defend why you're going to take your approach. You also want to carefully think about the limitations of your approach and consider alternative approaches. This is also a very common criticism of grants, in that there are many grants that don't consider any alternative approaches. They just have a main line of proposed experiments, and they think they're going to get results from them and going to be able to interpret them without any issues whatsoever. And so if you don't consider that you could have an issue crop up in your experimental approach, or have a secondary way of looking at that question to confirm, reviewers will key in on that. Another kind of deficiency is that you don't include sufficient details of the experimental approach. You don't need to tell them the millimolars of drugs or things that you're going to use, unless it's something really critical to the experiment. If you're using an inhibitor, and you might want to tell them the concentration you're going to use it at, but you really don't need to provide all of the scientific details of commonly used approaches. You can cite papers which you've done it, or other papers, but you need to provide them at least enough detail to understand the experimental setup broadly. You just don't need to tell them about every mouse, all right, or every human. And it's also really important to describe the statistical methods. This is brought up more and more by reviewers, that the statistical approach that will be applied is not described in any detail, or they're using the inappropriate statistical methods to analyze the data that they're going to generate. So make sure you pay attention to that and get help with the statistics if you need it. It's great to have a biostatistician involved in the work. And then, the other really common complaint is if a reviewer can't read anything on the figure in the preliminary data or the table that's provided because you've shrunk it down to a size that's ineligible, not legible for the readers. And so, you want to make sure that all of your figures are clearly able to be seen by all the reviewers. Try to keep them as simple as possible and as clear as possible, and don't shrink them to a size that nobody can read. Common issues with the specific aims are usually too ambitious. As I mentioned, I try to write just 2 or 3 aims. When you start going 4 or 5, I've seen a 6 aim grant. There's just zero detail in any of those. And so reviewers are not going to like that. Also, specific aims can be unfocused. Try to avoid that, or the goals of the aim are unclear. And so you want to make sure that the experiments that you're proposing are going to test your hypotheses that are detailed in the specific aims. And then sometimes we'll write specific aims that are a little more descriptive and limited in scope, and they're not really mechanistically focused. And you also want to make sure you articulate the future directions. It's great to have a well-written aim and everything, but nothing ends with the few experiments that you can write in that aim, right? You're going to be doing this work for 5 years. You want to make sure in each of those sections that you provide 1 or 2 sentences about what you're going to look at in the future as well, based on the results you get, to give the impression of what comes next after those initial results, right? And what you're going to follow up on. It's important for reviewers to have a sense of the direction you would head given a series of results. Common issues in the significance is that a reviewer will say that it won't advance the field sufficiently, and it's incremental. And this usually relates to the rationale not being compelling and well articulated. And then they'll often just write that the research is incremental, that you haven't advanced it enough for the field. Common innovation issues that crop up, again, does not advance the field. That can be in both significance and innovation. It's not conceptually innovative, which just means you haven't done a good enough job in telling the reviewers how your work is conceptually innovative for the field. And then the kind of most common one is it's not technically innovative, that you use common approaches. And then the common approach issues are that you have either too little detail or too much detail is provided. And this can be a common one for new investigators. Generally, you feel like you have to provide more detail because you may think you're not going to get the benefit of the doubt as a new investigator. And so you provide too much experimental detail at the cost of not providing enough rationale. And so you want to make sure you strike the right balance there. Also, a common issue with the approach is not enough preliminary data are provided to establish the feasibility of the aims. The experiments don't test the central hypothesis. And then again, the most common one I see is a lack of discussion of any potential pitfalls or alternative approaches. And then no description of how the results will be interpreted. I've seen grants written with just the experiments and then a very small description of what the interpretations of the experimental results will be. And generally those grants didn't fare well. And then some common ones with investigators. One is lack of expertise or publications in an area. If you're forging off into a new area that you don't have publications in, you want to make sure that you get a team of investigators to help you that are familiar with that area or those approaches. And you want to make sure you add those as collaborators. There really can never be enough collaborators on a grant. They just really help solidify things if you need scientific expertise in any area. And then, of course, having collaborators is great, but if none of them write letters for you, then that will be viewed negatively by the reviewers. So you want to make sure each of your collaborators are providing you a letter of support, detailing exactly what they're going to help you with. And that really helps the panelists. So just to wrap up, my best advice is really give yourself adequate time. I know that's really difficult. We're all crunched for time. We're all very busy teaching, doing research, helping our lab members. But you really need to think about the grant ahead of time and really the background rationale. Spend a lot of time on that. Make sure you have colleagues read your proposals. That's actually the best advice I can give you. They're the best source of advice you can get. But you can get conflicting advice when you have more than one colleague read your proposal. My best example of that is I had a 3-AIM grant that I gave to 3 of my colleagues, all that were close to the area of the grant. And each colleague came back wanting the aims in a different order. In basically the 3 combinations of orders that the aims could be in. And they all felt strongly that they should be in the order that they were telling me for, for very different reasons. And so at the end of the day, you have to pick when you get conflicting advice. It's ultimately your decision. But and then you just have to be persistent. Don't get down. We all get proposals that have what we deem as bad reviews. That's all of our experience, but you can't let that affect you, and you just need to be persistent. You need to pay attention to the criticisms that are raised by reviewers, but you need to get that grant back in. So, and with that, I'd be happy to stop and take any questions.
I got one question. This is maybe more about like the art of communication with the grant officials. So I'm working on influenza induced lung immunopathology, and then just with the hope of being assigned to an IC, which will have a better pain line. It's been difficult for me to come up with a strong enough justification to request my grant to be sent to the National Heart Lung Blood Institute. So I know that sometimes it probably doesn't really matter, but I would like to hear your advice whether or not it's how important is it, and then how hard should I try each time I submit a grant? Thank you.
Yep, so that's a great question. So actually, most of my grants get dual assigned to NIAID and heart-lung blood, because I also work on respiratory viruses. But they're generally primary in NIAID, and I know your question is related to how to get it primarily assigned to heart-lung blood. The best advice there I could say is try to look and contact the most relevant program officer in heart-lung blood and talk to them about what your research is and what you look at. And they can give you strategies, because the assignment is really about your abstract paragraph and what your grant is focused on. If your abstract is written in such a way that it's clear it's about the pathogen, it's going to get assigned to NIAID. But you can write it in such a way that it's more clear it's about lung damage, which NHLBI cares about a lot, right? And you can de-emphasize the pathogen if you want to try to get it to NHLBI. So those are ways in which you can try to get it assigned to one institute or the other. It's all about the grant title and that abstract paragraph, because that's all they actually use for assignments generally.
Yeah, I could not agree more with Steve. I actually had an R01 that was assigned to NIAID, but because it was actually a sort of a clinical trial, and NIAID does not support that for an R01, it was reassigned to NHLBI. So there's a couple of tricks how you can really guide it towards NHLBI and avoid NIAID. So if you make it as part of a lung disease, an exacerbation, let's say, of asthma or susceptible population like COPD and smokers, you go to NHLBI. So you're basically de-emphasizing the virus and using the virus to investigate a lung disease.
Thank you.
I have a question for Steve. So I asked several people about this, but I got also a mixed answer. Do you think that we should add reference to our specific AIM page? So I asked several people and I get a mixed, answer basically to that.
I've never. Yeah, actually, this relates to both references and sometimes figures, right? Some people will put figures in there too. I never do, because I feel I never want to sacrifice the words that I have, and I'm limited to one page, and
Right.
I think it's just too critical. Maybe I'm just not succinct enough to be able to sacrifice that, but I've never heard anyone complain that there's not a reference on a specific Ames page, or that they would need to see that. I don't think it's necessary nor warranted to include references on the Ames page. I see it very rarely, quite frankly. I don't see it as commonly used.
Interesting.
Yeah, just to follow up, I actually see it a little. I never used to see it, and I think NIH had like an instruction to not include it, which has, I think, been lifted. One thing that has now been implemented for NIH, I used to see sort of summary schematics of the overall specific aims on the Ames page, and you can no longer do that. So the specific Ames page has to be without figures and illustrations. That's a new one.
That's it. Oh, it's
Yeah.
interesting.
Yeah, so don't put it in there.
Is there any information on NIH page that's actually, reflect this, or?
Yeah, I mean, the long document that explains all their rules will have it, but
Okay.
yeah,
So,
they don't.
I would assume if they actually, if you have it on, you will be like, they will not review your grant, right?
Yeah,
No,
they could
it'll be
reject it.
kicked back.
They
Yeah,
reject.
it'll be kicked back. If you have a good Office of Sponsored Research program, they'll actually catch it,
I see. Interesting.
because they actually read the fine print of NIH instructions. I don't.
Yeah, ours will bounce stuff like, links that shouldn't be there
Yes.
on your
Yes.
BioSketch and everything, because a lot of people like to put links of all their papers and things, and you actually can't do that. You can have the one link, to your MyNCBI, but you're not allowed to put any individual links on the BioSketch page.
And anywhere else.
Yeah,
There's no more
you
links.
can't have
You
any
can't.
links inside the document.
Anything.
Grant. Yeah.
Even if they're not hyperlinks, you cannot have any URLs.
Yeah. So our office scans
Oh yeah.
very tightly for that.
Oh
So
yeah.
great question. So
Yeah.
If there aren't any more questions, we can move on to Dr. Jaspers' presentation. So Dr. Jaspers is a professor at the Department of Pediatrics, Microbiology, and Immunology, and the Department of Environmental Sciences and Engineering. She's at the University of North Carolina, Chapel Hill, and she'll be talking about this from the reviewer's point of view.
Well, thank you first of all for the invitation here, and thank you for Dr. Varga to actually give a great introduction. I think a lot of it was covered by him. So, but maybe I can sort of, and maybe with his help as well, we can sort of like provide a summary of what reviewers expect and what like works in my view, what I like to see and what really annoys me. So it's really more of an annoyance than rather not liking it. So what do reviewers expect? And that really depends. And it depends on a number of different factors. It really depends on what the funding agency wants. And this is really, really, really important. I mean, most of us probably sort of go towards NIH, but I think the question we had at the end of the previous talk already alludes to that. Like, look at what does the funding agency want? What does the institute want? If you're not going towards NIH, I just participated in a review panel for the Department of Defense, very different sort of outlines of what they want and what the goal is there. So make sure you understand what the agency you're targeting for your funding actually wants. Let me see what I can do this. Okay. So, and there's many different alphabet soup jargons here, and make yourself familiar with that. So there's an RFA, which is a very specific one-time solicitation. And so those usually do not go by a percentile, they just go by an impact scoring and a ranking, because you basically are competing to a very specific pool, and they fund maybe one, the top 5, the top 10, whatever the RFA wants. Then there are these funding opportunities announcements, which are FOAs. And that really is towards a sort of an initiative for a federal agency. These are usually the result of it's a discretionary award. So a lot of those, for example, came as part of COVID. So there were several different FOAs. Then there is what's called NOCs, and there's not really, depending necessarily, more money set aside for that, but those are called Notice of Special Interest. They're often sort of part of a potentially you can have as an administrative supplement to an existing award. We've gotten those for specific NOCs, and they're really a specific topic. A specific institute or a few institutes are interested in and supporting. Then there's the parent announcements. So those are the unsolicited investigator-initiated awards. Your run-of-the-mill R01, your run-of-the-mill R21. So those are parent announcements. And then there are PAs, so there are program announcements. And they're really used by an institute to propose as a research program in a particular area. And there's omnibus announcements. So, for example, there is program announcements in the context of Alzheimer's disease or specific areas within an institute. And they can be funded by set-aside funds. Those are PASs. Or PARs, which are actually reviewed by the institute rather than the scientific study sections that are normally convened for your unsolicited awards. So just make yourself aware of what it is that you're doing, who is potentially seeing your award. And what is an FOA name? And actually, this is basically what sort of sets this apart. So the first 2 to 3 letters are indicating the type, then the institute, the year of the publication, the fiscal year of the publication, the number, which is really in the order of the opportunities they occur throughout the year, usually a very brief title, and then the activity code. So it's either an R01, an R31, or an F31, or things like that. So that's basically what makes this title. So these are the components of a title. What is really, really important, and I cannot emphasize this enough, read the instructions of what you are, where are you putting, or what kind of application you're putting together. This is really, really important because every one of these, even R01s, and we just talked about this, the R01s are changing. Some of these instructions are changing. The page limits, I can tell you from awards that basically got kicked back because the page limit was not recognized. That is really annoying. You've put all of those work together, and instead of having 6 pages total, you basically have your specific aims page plus 6 pages, and it gets kicked back. Special components. As I said, I just reviewed grants for the Department of Defense, and there's special components that need to be included. So don't omit those because that basically kicks you out of the running. Budget limits, that's another one. For an R21, there's very, very specific budget limits. Don't go over that because again, those are also special requirements. That was actually, Dr. Varga sort of alluded to that. When you are referring to your collaborators, make sure you have a letter of support. Make sure that a reviewer can find those and knows exactly where they are. When you are putting together any kind of training grant or like an F grant or a mentor grant like a K grant, make sure you have all of your letters of recommendation and make sure that a reviewer knows where they are. And then, NIH always has tricks up their sleeve. They're changing the requirements constantly. They're changing the biosketch format constantly. I have no idea why they're doing it. It's not like there's new information in there, but they're now changing the way it needs to be laid out. It's super annoying, but if it's not in the right format, you're giving a reviewer basically food for critiquing you, or NIH actually kicks it back. So not including any of these special requirements is really annoying. I can tell you when I've reviewed grants and you're basically relying on a technology or on a reagent from someone and the letter of support is not included, that's really annoying. So make sure all of those are included. So we already talked a little bit about the specific components of the application, and I'm not going to go much into that. You already know about this. This was covered in the previous grant. Just want to sort of from a reviewer's perspective, for example, this is just the summary abstract, and I'll go into the narrative as well. Make sure you are highlighting, and for a reviewer, and also for the other members of the panel, because again, this is something that they all see. Make sure that it's easy to identify what you want them to see. The long-term goal, for example, the public health relevance, the field of the significance. Basically, make sure you have reviewers. Where's the innovation here? What's the central hypothesis? What are your specific aims? Make sure that the eye of anyone reading this on their screen is really guided towards what you want them to see. So make it really easy for people to identify the components you want them to see. The same thing is true for the narrative. Now, I actually, unfortunately, don't pay much attention to it. I think that's a mistake on my part, because that's really, it's not really something, a criteria, but it's really for the public health, because that actually gets published. So make sure you have the public health relevance and the potential to, any kind of impact it may have. So have yourself a sort of a little bit more of an abstract and narrative checklist. Make sure that your abstract, and this goes back to the comment that we just had about really guiding it towards a specific institute. This is really critical. Make sure your abstract has the keywords that are really appropriate for the institute you want this to go to, for the study section you want this to go to, and distinct enough to really avoid any confusion with any other terms. This is really important because we may think about certain terms in a very different way. So the research strategy that was already covered is really the meat of the application, has the significance, innovation, and approach. And they all must fit within the page limit. But this is really something more for a reviewer. Make sure you have a very clearly defined and easy to follow organizational structure of these components, and make sure that they are separated either visually by color or by different paragraphs or underlining, bolding, whatever you're preferring. So some people do significance, innovation, approach, and then do the preliminary studies or preliminary data first, and then they basically describe aims 1, 2, and 3. Another option is to have it by the specific aims and then the preliminary studies afterwards or ahead of everything else. So you basically, for each aim, you have a significance, and innovation, and approach. Or you basically have, for each aim, you have all of the things pertaining to that aim in your separate components. Everyone has a different approach, and you can have sort of hybrids of these approaches, but make it very clear and stick with that organizational structure. So it's really easy for a reviewer to see where they are in that on that particular page. Again, make it easy for the reviewer to follow. Tell me where I am within your application. Use color, use different sections, and in addition, have white space. Don't just have a page of nothing but words. That is really annoying. We already talked a little bit about this as well in terms of the significance. That's really a difficult criteria to score. I have actually somewhat of a hard time at times. Really sort of show what the importance, because the significance should be scored on not whether or not you can do this. It's irrelevant for the scoring. It basically, if everything works that you're proposing in your grant, what is the importance of the problem you're trying to solve, and what critical barriers are you breaking here? How can you improve the scientific knowledge, develop new scientific paradigms? What is the technical capabilities, the clinical practical implications here? new concepts, methods, treatments. Basically, this is not significant from a reviewer's perspective, should not critique whether or not you can do this. That's irrelevant for the significance. The significance should assume that everything you're proposing can be done. Again, make it easy for me to identify what the significance is. I cannot stress that enough. I don't want to find, what it is that this grant can actually do, what kind of importance, what kind of critical barriers, what kind of new paradigms, what kind of new treatments, new sort of preventive interventions can be done. Show me where it is, spell it out. I cannot really stress that enough here. So the approach getting started, and again, this was already covered in the previous talk. Describe key areas focusing on new things, on new data, highlighting, going back to the big picture, and really provide enough explanation and detail that really provide confidence for the reviewer that your idea is sound, that you can actually solve this problem, and that you have demonstrated feasibility. Proof of concept studies or data, even though they may not have any relevance to the exact research you're proposing to do, is really, really important to provide confidence for me as a reader that you can actually do this. This is a big one, and I cannot stress this enough. You need to provide sound statistics and data analysis plans. There's more and more omics, there's more and more sort of data integration, there's more and more data analytics in all of these grants now. This really determines the rigor of your experimental design, and that is a criteria that gets scored on. Provide a sound and detailed statistical analysis plan. Make sure you are addressing sex as a biological variable. If that is not in there, it's an easy target to address. It's not only for animals and clinical studies. If you're only using male mice, make sure you tell me why. If you're only using female mice, make sure you tell me why. But it's not only for that. If you are using ex vivo models of either cells or tissue obtained from mice or from humans, it still needs to be included, because we all know that cells and tissues remain or retain some of the sexual dimorphism that they have in vivo. So make sure you include that. Make sure you have a sound sample size. Again, this includes in vitro as well as animals. Don't tell me because we've used an N of 5 animals in the past. Show me what this is based on. Make sure you have a power calculation and a sample size, and have a sound statistical analysis plan. Don't tell me you're just going to do t-tests. If that is what's basically all you're going to do, that is no longer sufficient, because the data that we usually get these days is much more sophisticated, much more complex, that a t-test is no longer sufficient. So that needs to be included and needs to be paid attention to. So this is, I'm not going to go through all of this, but this is really common questions that reviewers want to see addressed in your approach section. Really, and this was already covered by Dr. Varga. What I want to drive home here from a reviewer's perspective is avoid alphabet soup, avoid jargon, and too much unnecessary detail. As Dr. Varga already said, I don't necessarily want to know how many microliters of PBS you're adding into, for an intratracheal instillation. That's not, don't save that space. What really annoys me too, and is very, very difficult to follow, is alphabet soup, where you basically are generating your own acronyms and you're using commonly accepted acronyms. You're using jargon that was not described. You're using a lot of very detailed mouse models with very fine changes because they have like one mutation, or they basically are inducible mice versus, whole knockout mice. Really try to avoid that kind of alphabet soup and jargon, because it's really, really difficult for a reviewer to then detect the nuances of the differences in your models, in your endpoints, in everything that you are using in your approach that may get lost. And it's really annoying if then the reviewer has to go back and always take a look at, like, what is this again? What is this a model again? If you have very sophisticated models and cell lines and mouse models, provide a table that is easy to go back to as a reviewer and say, oh, this is what this is. Okay, now I understand what this means. So really make it easy for anyone to follow. In the approach, again, this is something that I cannot stress enough. Most reviewers review grants in their quote unquote off time or spare time. This is not necessarily something that I do when my mind is the freshest at like 9 a.m. in the morning, because that's usually when I have meetings, I participate in other committees, or I don't have necessarily the time to read grants. So tell me a story that I can easily follow. So for example, when you write your research plan, and this is an easy strategy, provide a very good, study rationale. Why, what address, easy to follow. You're telling me what your research, what research gap you are addressing, what kind of hypothesis is driving it, what is really supporting it, what kind of data analysis plan you have, what kind of interpretation you have, and what kind of potential alternative strategies you're going to be using if your experiments are not working. And we all know that even though you can have a very detailed research plan, there's always things that are not working out. So make sure that you're aware of it. And what's really important here is that sometimes, a picture tells 1,000 words, and that's really, really true. I've seen throughout the seminars today that many of you use BioRender. BioRender and other sort of graphical scientific cliparts and graphical programs can really generate great schematics of your research strategy that really make it a lot easier to follow, and just keep it going, put it back in and sort of have a reviewer follow it through. It also provides a nice sort of point for a reviewer to take a breath. So if you just have words on the page, a colored schematic that really sort of summarizes what it is you're going to be doing in that specific aim is a much welcome change in that page, and keeps a reviewer engaged. So here are just some examples that we've used, sort of provide a visual overview of your aims. Again, this cannot be on your specific aims page, but can actually be at the very end of your background and significance. And I'll get to that in a second, why that actually is important. For those that have actually sort of write K grants or F grants, provide a timeline with the aims and the training integration, so that's really nicely color-coded and really easy to follow. Again, it breaks up the monotony of words, it breaks up the monotony of black and white, and actually is an eye catcher and draws the reviewer's eyes to a specific question. This is, you all know, you're all very familiar with the scoring system. A 1 is obviously the best score for NIH. You can get a 9 is a poor. And these are the sort of additional guidances as to what actually is a 1 and what actually is a 9. So this is when you go to NIH study section, this is something that is provided to every reviewer. So it's, it's pretty straightforward, what a 4 is, a strong but with numerous minor weaknesses, versus a 1 exceptionally strong with essentially no weaknesses. However, what is negligible, what is minor, what is moderate, or what is major? So that really is up to interpretation. So make sure that if you yourself recognize a potential weakness in your research or in your research approach, that you have a very good approach or very good alternative approach, so it never becomes moderate or major. So everyone knows that is a weakness. I can't do anything about it, but because they've addressed it, it becomes negligible or minor. So what are the review criteria? And that's basically, you get an overall impact score. And the other scoring criteria, such as significance, innovation, environment, investigator, all are scoring criteria, but everyone gives an overall impact. And so what NIH really says to reviewers is they will provide an overall impact score to reflect their assessment of the likelihood for the project to exert a sustained, powerful influence on the research field involved, basically have a meaningful and sustained impact. But what is the reality? If you go to study section, the approach and to some extent the significance really largely drive the overall impact score. And here's some data that was collected by NIH. So you can see the approach as well as the significance really drive the overall impact score. It is really more the exception that you have, let's say, an approach of 4 or 5 and an overall impact of 1 or 2. So this is really very unusual. You can have sort of like an overall approach of 3 and an overall impact score of 1 or 2, but that's sort of like the range where it's going. So the overall approach and the significance usually drive the overall impact score. The significance, what is that? So Webster basically tells us the quality of being important. And as I already told you, the significance is basically how will your work be, what kind of impact will it have on the field if it is successfully carried out? Now, the current NIH has actually modified that. Again, this is one of those things, so stay on top of it, is the impact based on the rigor of the prior research. So you have to have a scientific premise, you have to have really understand, defines the rigor based on the quality and the strength of the prior research used as the basis for the proposed research question of the project. So you really need to make sure that you understand what has been done in the literature, what has been accomplished by other investigators, and that really drives your significance now. We talked a little bit about the innovation. And again, this can be many different things. Very often, as Dr. Varga alluded to, people sort of look at the methods, technology, analysis, or approach as really sort of like the experimental innovation. But it can also be novel concepts or paradigms or mechanisms, possibly identifying novel therapeutic targets or potential novel intervention. It can also be a novel research team, and that's something that I've used. I have several MPI grants because I recognize I can only do so much, and I love working with people that are very much outside my field. And sort of like putting together a novel research team that has not previously worked together. And I'm not talking about necessarily, a geneticist and a biochemist. That's very, very close. But for example, if you have all of a sudden you're working with an organic lipid biochemist that basically has a very novel sort of skill set, and you're putting this with analysis of tissue, or even going further, you're going with engineering, and you're implicating that in your research team. So a research team can be very novel as well, but spell it out. I think making sure that what kind of innovation you're having, what kind of innovation you're proposing, and spell it out. Basically saying it has technological innovation, ... It proposes a novel paradigm, ... So basically spell it out, how it is innovative and what components are really strengthening the innovation score of your application. Investigators, as I alluded to, I have several multi-PI grants because I think science needs to be a team effort. But as that, you have to have an MPI plan, a multi-principal investigator plan. Make sure you have a well-developed one that actually addresses multiple different components. Let's say you have a fight, conflict resolution. You also have to have a good communication plan and things like that. So when you do that, make sure you include that as well. But I actually like to summarize the investigator component in the main part of my grant. And I do this usually in a very succinct paragraph because it provides the reviewer with sort of really making sure that all of the roles and all of the responsibilities of the different components of the grant are covered. Make sure you're saying, your statistical plan was reviewed by so-and-so. If you have a clinician co-investigator that provides sort of integration of your data, make sure that's in there. So you can really show in this particular paragraph that your team that you've assembled for your grant is bigger than the sum of its parts. And I think that's really, really important. In addition, it really saves the reviewer time to review biosketches. We all have to comment. We all have to write a comment in the investigator scoring criteria. And if you can save time to go painfully through the individual biosketches, that's really something that is reviewed favorably. So I usually put that in there as a small paragraph. Finish strong. This is really, really important. I can't tell you how many times I have read grants, again, where let's say you have 3 aims. Aim 1 is super strong, is really, really strong, very, very clear. And aim 2, yeah, is good. But then aim 3, it really showed the particular applicant ran out of energy. So finish strong. Include a timeline. I think it's really important that you've thought through how you are actually going to be doing this grant and what is the proposed timeline. For an R21, you got 2 years. How are you going to do the work that you proposed? Because very often R21s fail because they're too ambitious. If you have a 5-year R01, tell me how you're going to do it, when you're going to collect the samples, when you're going to analyze the samples, when you're going to write the papers. Show me what your timeline is. And then at the very end, I always put in, again, summarizing the impact of your proposed studies. We already talked about, Dr. Varga told you that at the bottom of your specific aims page, you want to tell them what the impact of the proposed studies is going to be. At the end of the grant, do that again to remind them, basically going back to the specific aims page. Okay, so this is the impact that this grant, that these proposed studies will have on public health, on this clinical issue, on whatever it is that you propose to do. To quote one of my former graduate professors, Dr. Arthur Penn, who is at LSU, he told me in a scientific presentation class I took with him probably around 1995, which now ages both of us. Tell him what you're going to tell him, then tell him, and then tell him what you told him. And that's exactly what this last paragraph will do, is summarize what it is. And it basically reminds the reviewer at the very end of your grant what it is that you just told them, why it is important, and how you're going to be doing it, and how it will really change the field. So with that, I wish all of you good luck, and I'm happy to take any questions.
I'd just like to say, Ilona, that he still tells them this.
Does he still have the horse slide?
I think so.
Oh,
I think
God.
I've seen it. Yeah, we.
Oh, God. I still remember the horse slide.
I could confirm he still has the horse slide.
He still has the horse slide. Oh my God. The horse slide, for those of you who have not taken his class, it basically is, it shows a horse, I think like jumping or something, and all it says is horse. And so the message he is trying to convey is really have a title that specifically tells more about what it is that you're going to say. It's just not basically saying results, right? Or ELISA data. So basically be more specific in your title. So that's what he said with the horse slide. So it's a legend.
All right. So so we tell investigators that they need to reach out to their program officers, but recently and that they have to have their specific aims page already finished at that point. So you really have to start way in advance. But one of the things I've been thinking about is those nosy statements. So if you're already funded, you get those emails and they're seeking information. So it occurs to me, and so I get that when I was department head, I would get those emails and I would start forwarding it to anybody it was relevant to, because that means something is coming.
Yep.
And have you ever gotten engaged in those conversations because they were seeking information, right? And before they compile a NOSI.
Well, that's an RFI.
Yes.
So an RFI is request for information. So that's where they want your input. The NOSI is a notice of special interest. So that's when they're already like ready to fund things. So the RFI,
yeah,
when it's just words, it's just feedback, it's just input.
yeah.
The
I've noticed,
NOSI
right.
is real funding.
But don't you think the NOSI comes as a result of
Yes.
yeah, so I think that more of us should get engaged in those things. I have not, but it seems like that might be a way to
Yes.
help our science, our fields.
Yes. Yes. So if there is an RFI, respond. Very often, some of these NOSIs are also just sort of organically developed, like COVID had a lot of NOSIs, or when eValley came around for, e-cigarettes, there was, so there's sort of like emerging clinical problems or a pandemic or something like that. Climate change will have, is just around, I guess it actually does have a NOSI. So very sort of logical sort of emerging health issues. But you're right. I mean, if we want to influence the field, we need to tell them what we think is important.
Yeah, I mean, you can't undersell what establishing a relationship with your program officer really can mean for you long term, right? And that's why we're talking about reaching out to them early and kind of often throughout the process, because if you just miss in an application, it's your program officer who champions trying to get your proposal funded, through any money that they may have left at the end of the year, which they didn't this year because of a variety of issues. But those program officers can give you information. They can help support you. If you ever need to apply for any extra money to either get a piece of equipment or support an underrepresented student, that's all going through your PO, your program officer. And so if they have a relationship with you, they're just much more apt and able to support whatever request you have. Some program officers will also give you heads up even before NOSIs are published that, be on the lookout for something, coming in the next few weeks or something. My program officer is actually very good about that because we have a very good relationship. So then obviously I kind of pay close attention to my NIH emails over the next few weeks that I might otherwise just file pretty rapidly. And so, those can be invaluable things, and it's really important to have an ongoing relationship with them. Also, if you publish a really nice paper, send it to your program officer. They want to know that stuff, and they will highlight NIH research sometimes, right? And the only reason that they do that is because people told them about it. So
yeah,
yes.
I think, Steve, that's a great idea. Sometimes if you publish some good papers, sometimes the POs also will, contact you. It happened to me a couple of times as well.
Yeah. So there's lots you can do to kind of help yourself long term by just building a rapport with that individual. And it's unfortunate they turn over a lot, and so the good ones end up getting promoted a lot. But you just never know, and they will pass on a good word to the person who's replacing them about you. So
Yes, thank you both for our very important talks.